Dun, dun, dun.
Hi, everybody. Welcome to the Biotensegrity party. And I'm Susan Lowell de Solorzano from Washington, D.C.
And I'm Chris Morita Clancy from Deep Cove in Vancouver, Canada.
And I'm Graham Scarr from Nottingham in the UK. It is our intention to promote and share the Biotensegrity concept with as many people as possible and to include you all in that mission. The Biotensegrity Party is an all-volunteer production of the Stephen M. Levin Biotensegrity Archive, a not-for-profit organisation whose mission is to educate, raise public awareness and build community in the field of biotensegrity, and to foster and forward discovery, research and understanding in the fields of science, health and medicine.
So we have our team today for volunteers. Thanks, Graham, by the way. We've got Mariana Barreto from Mississauga, Ontario. How'd I do, Mariana?
Hello, everyone. Good, thank you. Happy.
Did I say it right for the first time ever?
You
Oh
did.
my gosh. And Gregory Chudy from Lancaster, or Lancaster, Pennsylvania, as they say it there. Hey, Gregory.
Hey, everybody.
Lisa and Rachel will be back next week. They're doing a integrative biotensegrity weekend for movement teachers, and we hear it's going really well. So today we have our special guest. We call her EChO, but her formal name is Dr. Eleanor Ory, and she does work at University of Maryland Medical Center in breast tumor cells and the microtentacles that form in them. I don't want to try to characterize her research, but I'm going to pass it over to you, EChO, for the toast.
All right. I want to first and foremost thank you all for having me and taking an interest in my work to spend your afternoon here. The focus is on cancer. I know a lot of people have lost people to cancer, and the goal of this work ultimately is so that less people suffer from this disease. So I thought I would dedicate the toast today to no more a cancer-free world.
There we go.
Cheers.
Cheers.
And
before EChO starts her presentation and we do a little sort of interview, I wonder if you could tell us about our sponsors.
Sure, thank you, Susan. Our Biotensegrity Party is an all-volunteer production of the Stephen M. Levin Biotensegrity Archive and made possible through the generous support of our sponsors. Handspring Publishing in East Lothian, Scotland, have a wide range of authors and books available for movement and manual therapy professionals. They're offering a generous discount coupon to those on the Biotensegrity Archive mailing list. Handspring also produce a regular newsletter, which is free, and you can join from their website, provides details of their ongoing webinar series with Elizabeth Larkham. Embodied Biotensegrity with Chris Clancy in Vancouver, British Columbia, who provide an excellent resource for those of you looking to learn more about biotensegrity. Chris is currently offering a 14-day free trial for her ongoing course, Biotensegrity Trailblazers. So check out the chat for more information. Then we have Integrated Biotensegrity in Alberta, Canada, who are holding a 3-day movement focus event on April the 24th to 26th, with keynote speakers John Sharkey, Joanne Avison, Wilbur Kelsick, and other presenters. There's also a discount coupon in the chat.
But Graham, I'm going to interrupt to say that's actually happening right now, February. So sorry about the date mix up. We didn't mean to give you the information wrong. I don't know if they can still join it, but I know there are a lot of people here today who are jumping back and forth between the two events. Thank you.
I believe, yes, you can still join, and you'll be able to even join after the fact to watch the recordings. Before we move on, can I please let everybody here on Zoom know we're taking questions in the chat. I'm going to be monitoring that. And if you're with us on YouTube, hi, thanks for joining us. You can always sign up for the Biotensegrity Archive newsletter And that way on Wednesdays, you'll get into your email inbox the registration link, and you can hop over and join us on Zoom next week. Meanwhile, Mariana will be at YouTube monitoring the chat there and transferring any questions for EChO over to our chat here. So thank
And
you.
I think that that date, though, April 23rd, 24, 25, is important because that's the International Biotensegrity Congress that's coming up. I think we've got a, Gregory was able to find a link for that, and that will be in the chat.
Yes, too many scribblings on my bit of paper here, that's why.
Well, but you know, Graham, I'll tell you what I think is great is we are now at the point where there are so many exciting biotensegrity oriented events happening that it's getting confusing. It's wonderful.
Indeed, yes, indeed.
So EChO, let's go over to you. I just want everyone to know a little bit about you and your journey to where you are today, including the fact that you're an aerialist and a contortionist, and not everybody who comes on to present the scientific research also is, you know, well established in the movement world the way you are.
Thank you. So yeah, I have done aerial pretty much as long as I've been out of college. So basically as long as I've been doing research. And it's wonderful to have that space outside of the lab bench to kind of explore.
Right, because you've taught trapeze, is that right?
Yes, I until the pandemic, I was teaching trapeze and fabric. Fabric is my bigger focus, and I used to do more net. And I really love it. There is a wonderful Biotensegrity model in its own right.
To be working with these fabrics and the trapeze and the tension, you and I did get to talk about that when we did some of our pre-interview. How did you get into cancer research so that we have the context for your presentation today? And I meant to say that Eleanor is going to be speaking a lot about her really breakthrough work that was published this past year in the Journal of Physical Biology. Is that right, EChO? Sorry, I'm trying to
yeah,
EChO.
2 years ago, but yes.
Okay.
I think it was 2 years ago. Yeah. So I got started after undergrad at NIH. I did my undergraduate work at Smith College with a focus in engineering and philosophy. I was initially interested in more neuro applications as well as artificial intelligence. But I really liked more of the mechanical aspects of cancer. And when I got to NIH, the project that I really enjoyed working with, I had a great team, was focused more in cancer. So I ended up going in that direction and continuing it in grad school and now my postdoc.
That's great. And I also spent many of my formative years at in the Happy Valley in Amherst, Massachusetts at Hampshire College. And I took a number of classes at Smith, and you were part of a pioneering program at Smith. I just want you to mention it quickly.
Oh yes, so Smith is an all-women's college. And we were the first all-women's college to have an engineering program. And I was the third class of that engineering program. And that was a really exciting, amazing thing to be a part of.
And isn't it amazing that it took that long in our human history for that to happen? But thank goodness it finally did. Well, if you're ready, why don't we just jump into your presentation then?
Totally. Let me share screen. And beginning. And you guys, I'm just gonna do a quick test. You guys can see that transition?
We can.
All right. So as Susan already said, I'm EChO. That is the shortened version that comes from my full name, Eleanor Claire Higgins Ory. I'm at the University of Maryland School of Medicine in the Marlene and Stuart Greenbaum Comprehensive Cancer Center. And they have been a really wonderful environment to do research. I enjoy being there very much. And today I'm going to talk to you about microtentacles, cellular mechanobiology, and kind of pose a question to you about what you guys think, being experts in biotensegrity. I have it categorized as quasi-biotensegrity, but I'm really looking forward to you guys' thoughts and input. Like I said before, ultimately the goal of this research is to treat cancer. So I'm going to start with a little bit of background about cancer. Some of you may be aware, like have relatives with breast cancer or liver cancer. And when we call something, say, breast cancer, we're referring to the primary tumor. That's the place where it originated. And so this is, as cancer progresses and becomes more aggressive, this is called the metastatic cascade. So after it starts as the primary, it starts invading surrounding tissue. And this step is called invasion. And then it next goes into the bloodstream, and that process of breaking the cells in the blood vessel wall and going into the bloodstream is called intravasation. And then it floats in the bloodstream, and this is what we call a circulating tumor cell. Ultimately, it reattaches, it extravasates into a new area and forms a micrometastasis or distant tumor. And this is a more advanced stage of the disease. And when you see that someone gets treatment for cancer, the vast majority of it is on the primary tumor. In fact, 95% of cancer funding is focused on the primary. However, 90% of fatalities from cancer happened once it metastasizes. And that's where fatality. So there's this whole area in between here. And within the metastatic cascade, a lot of people focus on invasion and migration. And that's the steps we talked about. But one other interesting fact is that even for cancer cells, the majority of them die in circulation. So both this, critical step in the metastatic cascade, as well as the really unique mechanical properties of being in a suspended environment, of being a free floating cell in the bloodstream, and how it ultimately reattaches. This is the step that I'm most interested in. And when I was a graduate student, I met my mentor, Stuart Martin, and he had discovered this these protrusions called microtentacles. And he had shown that when you put them on a plate with endothelial cells are the cells that line our blood vessels, that it reattaches. And then they looked at many different genetic changes and drug treatments, and they found like there was a high correlation between cells with a high microtentacle phenotype and reattachment as measured also by impedance measurement. So like when you have more cells on a surface and you put an electrical, make an electrical measurement across it, it changes the impedance. And we wondered if this was a plausible mechanism for circulating tumor cells. And the reason this is being shared in a Biotensegrity forum is microtentacles tend to form when there's unbalanced forces between the actomyosin cortex and the microtubules pushing out. And we'll go into more detail about that in a moment. I do want to kind of review a little like what has been my understanding is of what a classical tensegrity model versus a quasi-tensegrity model. And feel free to give feedback on that in the end. But you have the floating compressive members that don't touch. They have uniaxial forces. So they're only being pushed and pulled in one direction. The cables are not supposed to stretch, and the compressive members are supposed to be exclusively in compression, and the tensile ones exclusively in tension. Whereas in the quasi-tensegrity model, we have deformable compressive members, stretchable tensile members, and you have deformable tensegrity. Oops. It's not going to the next slide.
It's a really nice slide, Eleanor.
Thank you. And those are my homemade models that I've started making since Susan's class. The engineer who really brought tensegrity into the cell biology world was Donald Ingber. And he was thinking more about attached cells, but he described the microtubules as in compression and the actomyosin cortex in tension, allowing it to be a deformable structure. And the actomyosin cortex to act as nonlinear semi-flexible chains. And he also included the substrate stiffness, like the integrins attaching to the ECM. And there are both compressive and tensile members in the structure that are dynamic. You have the microtubules growing and fracturing, as well as with the actin filaments and myosin constantly moving those filaments.
EChO, before you move on from that slide, the ECM is the extracellular matrix, is that correct?
Yes, extracellular matrix.
And then MT disruption.
MT is microtubules.
Okay.
And the nice thing about our models, what we're looking at, is it's in suspension. And in some ways that allows a more pure form of a tensegrity model, because we don't have to think about the anchoring of the cell to other cells or the extracellular matrix.
Great, thank you.
So let's talk a little bit about the actomyosin cortex from like a global perspective. I represent it here as kind of this noodly network. It contracts inward. And there are this network, just to give you kind of a sense of the scale. The dark space in between the little like holes in the actin filament network can range from say 20 nanometers to 200 nanometers. You also have the microtubules, which are 25 nanometers in diameter. However, some really beautiful work done at UC Berkeley by Kevin Downing, who is sadly no longer with us. This is a electron microscopy. And he showed that the microtubules bundle together in these microtentacles. So this is treated with a drug that we'll talk about a little bit later. And this is a more longitudinal view where you can see the individual filaments. So kind of zooming in, actomyosin, there's a lot of things, different filaments and intermediary filaments that are considered a part of this cortex. But the ones that I'm really going to focus on for this talk are the actin filaments, that are these long things, and the myosin heads. And the myosin, it has these two heads on each end of it. And it pulls the filaments together, and that's what gives us that global contractile sense. So talking a little bit more about the drugs, because this is a force balance system. In our normal cell, we have the this is the green, these arrows pointing from the center out are microtubules, and they are in compression. And then in the red on the border here, we have the actomyosin cortex, and that is pulling in. And in a normal cell, depending on what cell type, because more aggressive cancer cell types tend to have more microtentacles, might have a few or none. If we treat that with a drug that disassociates all the microtubules, we lose our microtentacle phenotype. However, if we use a drug like paclitaxel, we get stabilized microtubules, and we end up getting a more microtentacle phenotype. On the other side of the equation, that's like where the actomyosin is pulling in. We have a drug called ML7. This basically prevents, it keeps the myosin head stuck to the actin filaments, and we end up with a very stiff actomyosin cortex, and we lose our microtentacles. And then if we want to disrupt the actin, we can use drugs like latrunculin, blebistatin, Y2762, and we make this softer, and then the microtubules can push further out, and we also get more microtentacles. And there's genetic changes that we can do to manipulate these two, but that's not the focus of this talk. We're not fully clear on why microtentacles aid reattachment. So my two kind of hypotheses is maybe, having more flexible microtentacles, maybe that'll help with the initiation, or maybe, having, once it gets attached, having microtentacles helps keep it attached so that the flow of the bloodstream makes it harder to remove it. And at this point, that question is not really answered. So when I started working on this project, the way we measured how much microtentacle or microtentacliness a cell or cell population has. So you'd have someone who didn't know the conditions, like the genetics or the drug, and you'd give them samples and they would count through a microscope with a little county clicker and determine how many had a microtentacle phenotype and how many did not. And so it was an expression. The other thing to talk about, and part of the reason why people don't like to study cells in the bloodstream, is they are hard to image. They float away, they don't stick. So when you try an image of free-floating cell in time, when you do like a time lapse, you can see it moves.
Eleanor?
Yes?
Echo, sorry. I don't know if you want to answer this now or later, but I will just give you the question from Graham Scar. How do microtentacles differ from filopodia and lamella?
Yes, we get this question a lot, even from cell biologists. Filopodia are actin-based. They're in attached cells. So this phenomena happens only in suspended cells, but microtentacles are tubulin-based and filopodia are actin-based. And in fact, when we first saw this, we thought they were filopodia. So we hit them with a drug that would remove actin, and then we got even more microtentacles, and we were like, what is going on? And that is part of actually how we came to discover this. And we're a little critical sometimes of other scientists about whether or not they're actually looking at filopodia or microtentacles.
Wow. And the lamellipodia is the other one that Graham's asking about.
Lamellipodia is kind of a ruffle of the cell membrane. It has actin. It is also actin-based. So like filopodia, I think it's more of kind of a morphological descriptor.
Great. Thank you. That's great to get that clarified. Thank you.
No problem. Thank you. We developed a tethering technique where we did these layers of polyelectric. And these tend to repel the inside of the cell so that it won't stick. But then to confuse the cell further, we put a layer of DOTAP, which binds to the lipid bilayer. And so it is kind of repelled by the surface, but also held by this lipid monolayer to the cell's bilayer, and it keeps it from floating away.
So like a helium balloon in your hand.
Yes. So we call this a tethering technique. Although it might be more like holding it at the base of the balloon. There are actually methods where they take like a strand of DNA and like physically, but it's so laborious that we're doing a lot of studies. It's not great. So when we get these tethered cells, you can see how nicely it doesn't move. And that's a time lapse, and that's a little tracking of the centroid of the cell body. And you see it doesn't move, but it also has not spread out the cell. So it still has this suspended phenotype. But now, if we want to talk more about like how many microtentacles, how long it is, we can look at it and say that, but we as scientists, we really like to measure things. We like to see numbers. And so that's where I started to come into this project, and I developed an analysis method using a rotating anisotropic filter. I was able to pick out these microtentacles, whereas a lot of imaging analysis techniques, we lose the tentacleness. And then I did a more traditional image analysis approach, and I got kind of what I call the more globular features. And then sometimes this ends up being a lot thinner, and it has kind of a crescent shape or annular. So I add in a very like crude cell center in. And as a result of adding those three together, I get an outline of the full cell, including the microtentacles. And then from max curvature, I can identify the tips of each one. And then using a older analysis method, I find what the cell body. So when I'm referring to the cell body in this presentation, I'm referring to the cell not including the microtentacles. So the microtentacles are protrusions beyond the cell body, and we kind of stole that terminology from neurobiology. So my first question was started with targeting the tubulin side of the equation and to see if that changes the dynamics. So we treated cells with vehicle. So when we add a drug to cells, we have to dissolve it in a solution that's usually DMSO. So for our control, we add DMSO so that we're making the distinction that it's not from the substance that it's dissolved in instead of the drug. And we're comparing apples to apples instead of apples to oranges. The first drug we worked with was paclitaxel, and this stabilizes the tubulin, makes it less likely to dissociate, so you have longer, stronger filaments. It also increases bundling. And then we have colchicine, which dissociates the microtubules and should give us less microtentacles. And below, oops, we have example outlines of each. And this is, these are stained with cell mask orange. It's a membrane dye. And in this first column, you have like the original image over a Z-stack, the max intensity over that stack. We can see the cell body, and it's not necessarily perfect. As you can see in the tubulin. And in the colchicine, it tends to get the sort of blebs. And then we have the outline for vehicle. You can see this is a 436 cell, which is an aggressive cancer type. So it already has microtentacles. And then treated with paclitaxel, we get a lot more microtentacles. And then with the colchicine, it's covering the full outline. And you can see then when we find the tips, we have some for the vehicle. We appear to have a lot more for the Taxol treated, and very few, really picking up mostly noise here with the colchicine. And this is what that looks like in video form, just to give a sense. And what interested me is it looks like the control microtentacles are moving around a lot more than the Taxol. But first, I wanted to look at the numbers. And we actually see that Taxol did not necessarily significantly increase the number of tips, but it did increase the distance of the tip from the cell body. So these are longer microtentacles. We can look at number and tips, but to some extent, I also wanted a measurement of like how microtentacally, like how much microtentacleness as a whole it has. So I came up with these two sort of composite metrics, and one I call the cumulative tip distance. And what we do to get the tip distance is we take the distance from the tip to the cell body for each tip, and we add them all together. So if it's longer and there are more of them, we get a higher number. And this sort of tries to account for both the length and number in one metric. And then as another metric, we took the ratio of the full cell to the cell body and got our measurement this way. And as you can see, when we compare that, we have an increase that's significant in cumulative tip distance for Taxol compared to control, decrease for colchicine, and also the same trend for the ratio. So I showed you a little bit on the slide above that when you're looking at the vehicle and Taxol, it looks like there's more motion. So I looked at these at a time series, try and see how much movement up and down, right? And one thing we in the more physical sciences like to do is a thing called autocorrelation functions. So what I'm looking at is as the cell is moving in time, I'm looking at it at one time point compared to the other. So if I compare right now to instantaneously now, there is no change no matter what. But 10 seconds from now, maybe I've changed only a little bit, or maybe I've changed a lot. So I compare 10 seconds apart for the vehicle, all instances of 10 seconds apart, and then all instances of 20 seconds apart, 30 seconds apart, etc. I do the same for paclitaxel. You can see it looks like there's less movements, especially for things 10 seconds apart. And yes, for autocorrelation, you can see this really steep slope for the vehicle, suggesting that there is more motion. And for the Taxol, it's a more gradual slope, or smaller decay of the autocorrelation function is what we like to call that. And so that suggests less motion. So that was just one cell. Now we look at it across a population of cells, and we see significantly that the Taxol moves less than the vehicle. And that's how we measure that motion. And so I'm going to summarize the tubulin side of this drug experiment set of things. And first, we showed that tethering allows the visualization, and we can better analyze the microtentacles than like just having it float in a petri dish. And we showed that Taxol significantly increases the distance of the microtentacles from the cell body, not necessarily the number. Colchicine significantly decreases the distance of the microtentacle tips from the cell body. Colchicine and Taxol are more correlated, suggesting that there's less motion. So then I looked at it from the actomyosin cortex, and this work is not yet published. So here it is. And I'm just going to talk about the drugs that I used. I went a little drug crazy with these cells. We have again our normal epithelial cell, our cancer cell. For our actomyosin disruption, we had latrunculin, blebbistatin Y27632. These soften the cortex. And for stabilization, we have ML7 and 4-chlorpheneran. And all of these drugs only impact the actomyosin cortex. Zooming in a little bit more, like what these drugs do closer up. In the normal, there's nothing going on. So we have that the myosin head binding and unbinding to filaments as it normally will. With latrunculin, we break apart the actin filament completely. They just disappear. With the blebbistatin, the drug binds to the head of the myosin, and so it can't bind to the filament and pull them in. Y27632 works with the ROCK pathway, and I'm not going to get into too much detail with that, but what ultimately happens is we end up with more actin filaments cut up, and we end up with the myosin not phosphorylating, so it can't bind to the head, or it stays phosphorylated. With the ML7, this also targets the light chain, which impacts its phosphorylation, inhibiting it, and this time it gets stuck in this bound position to the actin filament. And then 4-chlorpheneran, 4-chlorpheneran basically prevents the actomyosin from making holes by inhibiting what we call septin. So because in this set of experiments, we started fixing things. That's kind of when we like kill them, but preserve them in their state. But that means we can do this cool thing called immunofluorescence. So we take antibodies and we can target certain proteins and then attach a dye to that so that we can visualize them. So we have a stain called DAPI. This binds to cells in the nucleus. And we can look at that across vehicle, latrunculin, blebbistatin, Y27632, ML7, and 4-chlorpheneran. We see like there's not necessarily a big morphology change in those. Then we stain alpha tubulin, and that lets us see the tubulin. And you can see like these nice long protrusions. This is how we kind of confirm that these are tubulin-based and not actin-based. And you see them in the latrunculin, the blebbistatin, the Y27632, and not so much in the ML7 or 4-chlorpheneran. Then we also stain the actin. This is a normal actin activity. We see that it's almost completely gone in the latrunculin. It's pretty dispersed in the blebbistatin and Y27632. And it is kind of concentrated in the ML7, and it's not quite sure what's going on with the 4-chlorpheneran. And then in the last row, we've colored each of them to kind of allow you to see them overlaid, where the blue is DAPI, the green is alpha tubulin, and the red is alpha actin. But for the image analysis, I use a stain called WTA. And that also stains the membrane. And you can see similar morphologies as we saw in the immunofluorescence. The vehicle, we have latrunculin with these crazy protrusions, the blebistatin with protrusions, Y27 with protrusions, this ML7 with no protrusions, and for chlorphenerin, there are some protrusions. It's kind of hit or miss with that drug. So using the same quantification methods as I used above, we have the number of tips or microtentacle tips, and we see increases for blebistatin and Y27632, not so much for latrunculin. And we see a decrease in ML7 and not really any significant difference for chlorphenerin, but you kind of see these two populations. So there's a lot more lower here. So like I said, it's kind of hit or miss, but it's not significant across the whole population. Then we have the distance of the microtentacle tips, and that's significant for latrunculin, blebistatin, Y27. It's an increase. ML7 is less, as we would expect. No significance for chlorphenerin. Cumulative tip distance, significance for latrunculin, blebistatin, Y2762. ML7 is less, and the same trend for the ratio. So those were with fixed cells. We wanted to compare them for live cells to do the dynamic motion. We can't do that with fixed. It's a little harder to measure with the actomyosin changes because they tend to be more dynamic, but we see increases for blebistatin Y27632 compared to vehicle and the number, not any significance for tip distance or cumulative and ratio. However, when we do the autocorrelation functions, we do see a decrease in autocorrelation for Y27632 in the number. Blebistatin Y27 in the cumulative tip distance, and blebistatin in the ratio. And this is suggesting that there is more motion. So just to summarize the actomyosin drug experiments, we get that the latrunculin increased length, both length and both of the composite metrics for microtentacles. Blebistatin Y27 and ML7 do as we would predict across all the metrics. Although it seems to be maybe that the number is a more significant criteria for these drug treatments. And then we have ML7 reducing metrics. And then we have blebistatin Y27 decrease autocorrelation for number of tips, suggesting an increase in motion. And this supports the force balance hypothesis. So kind of close to summarizing with whether or not the microtentacles fit a Tensegrity model. When we look at the classical Tensegrity model, maybe not so much. One of the criteria is floating compression. So do the compression members touch? So no, it doesn't fit that model because the microtubules meet at a microtubulized organizing center. Uniaxial forces, no, because the cytoskeleton is really experiencing like fluidic shear forces, which the stable structure, no, they're constantly growing and collapsing and associating and dissociating. So it's not really a stable structure at all. And the myosin is also constantly associating and dissociating from actin filaments. The cables do not stretch, and tensile members are continuous. No, actin has kind of this branching network, so it's not really continuous. Are the compressive members exclusively in compression? No. The literature has a lot of cases where they show that actually microtubules do go in tension. But it does seem to fit these criteria for quasi-tensegrity of deformable compressive mesh members, stretchable tensile members, and deformable tensegrity. And then this last slide I wanted to show you because I talked a bit about whether we thought microtentacles might help with initiating or strong attachment. But a thing we've learned kind of later on as this project goes along is when you look in circulating tumor cells in actual patients, the ones with the worst prognosis, it's not just a single cell, it's a cluster of cells. And this is generally really a bad indicator for cancer patients. So what I did is I did a clustering assay and with the actomyosin drugs and compared. And interestingly, with these drugs across experiments, we see an increase in clustering compared to the vehicle. So maybe microtentacle, at least for the actomyosin cortex, assists clustering. And on the bottom, I just have sample images of what that clustering assay looks at. And with that, I want to say thank you and let me know if you have any questions. These are my lab members, my mentor, Stuart Martin. I really love working with my lab. They're a wonderful group. I also must thank the American Cancer Society because they are the one that pay my salary and let me spend my days doing cancer research. And with that, I will open up. Oh, and you can reach me at this email address, and that's my Twitter handle.
Great. Eleanor, before we open it up, generally, I think I'm going to want you to put that slide back up in a minute that has the, where you were saying, does it match this? Yes, no. And for the conventional tensegrity model. The other thing is, I want to know, so I've been teaching a class based on my Biotensegrity book through Chris's embodied Biotensegrity platform. ECHO's been a part of that. She's also been going wild with the model building and the exploring and really applying her engineering acumen to that. ECHO, I'm wondering if you remember when you first encountered a tensegrity model.
Like a physical, like build one with my hands.
Yep.
A few weeks ago. Like, no, I never built one. I have my cells in the lab.
Right. Yeah. So that's one question I have. And how common do you think it is that people who are working with the tensegrity model or, alongside the tensegrity model or somewhat in that context at the cellular level, how common do you think it is for them to ever have held a physical tensegrity model?
Not very common.
And so you're
I mean, also understand we're a niche area. Only recently have other labs started looking at microtentacles. Like I mentioned earlier in the study, the majority of metastatic research focuses more on like the migration aspects.
Right.
Although CTCs has become kind of a hot area.
And so and I know when you were in Baltimore recently, you took a venture.
Yes.
Can you just talk about that?
Yeah, I meant to put some of those pictures in this presentation and totally forgot. Yeah, because that is where my lab is. It's like a 10-minute walk from where my lab is. And after reading part of Susan's books, I'm like, oh, I can go see that. And in a day I had, because we have to get tested for COVID regularly, and I was already near the convention center, I took a look at that. And it was a neat thing to see.
So you went over to the Science Center and you saw Snelson's Easy Landing.
Yes.
So that was the first time. Was that the first time you remember seeing or noticing this large tensegrity model?
I think I had noticed it before. In my engineering, I think at one point as like sort of a interesting thing, we might have briefly mentioned tensegrity models and like maybe our like intro classes. I didn't really like break one down and study the mechanical forces at point in time. But that's probably the first time I was like exposed to the existence of tensegrity models.
Right, right. And so now that you've, held one in your hand, you've built one, you've gone and seen this huge sort of networked one, interconnected one that Snelson did. How does that influence your kind of framework or sensibility when you do your work? Is it something you would recommend to other people who are working in your area?
Yeah, I mean, I think it's always, I mean, I'm a kinetic learner, so I always like to be tactile, and I think a lot of other people benefit from that too. I would love to see a more handheld model that might closer approximate the cell. And that's kind of something I'm thinking, like, how can I build something like this that is simple and might be more similar to like the microtentacles, because I can't really make those protrusions go kind of beyond the system without really breaking it. I mean, the really loose cables and sort of the classic string and popsicle stick one, if I loosen those kind of, but then it kind of gets flat. So yeah, it's something I've been thinking about.
Good. I mean, I have to say, for me studying this, I'm really a slow learner. Things kind of have to sit, and then I touch things and move things, and then they sit for longer. And, I'm typically the last one to put it together for a lot of information. I will just say, so I completely understand, and everybody learns differently.
And I mean, it was a useful, I mean, even though it didn't fit the classical tensegrity model, it was very like having that as a starting point and thinking about whether it's impacting the actomyosin versus the tubulin did help us organize that information in a simple and elegant way, I think.
Yeah.
So it's useful. It was still useful, even if it doesn't fit the classical model.
Okay, that's great. Thank you. So now I think, if you don't mind, I'd love for you to put that slide up, and Steve, I think, wants to chime in. Could somebody make sure Steve's unmuted? And I just want to say, if you can put that slide up, you know which one I'm talking about.
Yeah.
We were chatting about this yesterday, and you can do a share screen on it or whatever. I think the value of your slide, and sorry, Steve, I just want to say one quick thing.
Go ahead.
I think the value of your slide here is that it is showing us how it is giving us insight into how the Biotensegrity model is being perceived largely in your field. And this is by you. You are better informed than most of your peers on both Tensegrity and Biotensegrity. And with that orientation, where you are in your process, these are the questions you're asking, which I think are excellent questions. And these are the answers you're seeing, which I completely understand. Can you make it full screen, EChO? I don't know.
Sure.
Maybe you can. And I'm going to turn it over to you with that. Yeah. Yep. And Steve, is this around the area you were thinking of talking, or? Oh, and one more thing. EChO, I want to say thank you. You are doing incredible work. I was near to tears. I am bowled over by your dedication and just how you are so fully applying yourself to figuring this problem out. I'm just so grateful. So over to you, Steve.
Well, hello, everybody. I agree with Susan that that was remarkable, the whole way it was presented. I was trained before there was electron microscopy, so all this is just fascinating stuff to me. But I have always thought down at this level. I have disagreed with Ingberg's model because I do not see tensegrities as ever shearing, that there is no such thing, and that tensegrity either expands or contracts auxetically. And the other thing I wanted to point out is that in our concept of tensegrity, the compression elements and the tension elements are reversible. There is no one that is always in compression and always in tension. So that we don't see it quite as rigidly boxed in as Ingberg has put it out. So I think there's room for the tensegrity model to fit into this work that you're doing. And also the microtentacles themselves, we see as tensegrity towers. So that because it's always a heterarchical system, so that within each structure, there is itself another tensegrity. They just nest one within the other. So we never see, so we always see at any point where there is a shear force, that shear force will be converted into a tensegrity structure where there is no shear force because it's much more energy efficient. So from the standpoint of the tensegrity model, I think we have to upgrade that model because I think it's been promulgated by Ingberg that, and there is no shear in tensegrities, and there is no distortion model in tensegrity.
Could I ask, thank you, Steve. Mariana Barreto, I hate to put you on the spot, but I would love for you to share your screen, and then EChO, we'll come back to maybe this if we need to later. But I'd like, and Mariana, I want you to show everybody the model that you made with the pelvis and the legs. Can we spotlight Mariana so she can see it? And here's what I was thinking. Okay, now turn that upside down and now move it up higher or back from the screen more.
Hold on.
Yeah.
I wasn't prepared for this.
No, of course you weren't. But hey, this is how I roll, my friend. We're improvisational. So when she was constructing this, she was saying, here's the torso, here are the legs, and you can put it that way, right? And she was having it walk. But now what I'm thinking is, if now you turn upside down, here's a branching microtentacle. And again, we're just looking at a moment in time in a frozen, because of course we're soft matter, everything's transmutable. There's all that stuff going on, but at a moment of time and at this one scale level, because all of those sticks and strings would also be Tensegrity towers, and you have to envision it that way. But here is a branching microtentacle. What do you think, Steve?
Absolutely. I mean, that's exactly what we're saying. When you look at the hot thing today is the coronavirus. I mean, those spikes coming off that virus are themselves Tensegrity towers. And they would be the same kind of things that are going on. And I would see that maybe the mechanism of a cancer cell is very similar to the mechanism of a virus hooking into a cell with some sort of receptor site.
Interestingly, those are also tubulin based on the COVID virus.
On the coronavirus, the little things, what are they called? The corona.
Just spikes.
The spikes.
Well,
all right, thanks, Mariana. You can stop the screen sharing. I think Graham's got a few things to say too, and I think we can invite everybody to turn their cameras on, and we'll kind of start sliding into the less formal part of the hour. But I do want to give, I know that I wanted to give Steve a chance to speak, and I know Graham, and I'm guessing, I think I saw Neil here, and I'd sort of like him to weigh in as well. Graham, go ahead.
Thank you, Susan. And thank you so much, EChO. This is really fascinating because I've not come across these microtentacles before. So it's a great presentation. I think you sell yourself short with regard to Tensegrity. I think this explains Tensegrity wonderfully. It's just that the models we use are models to help us understand different aspects of it. So when Ingber described the microfilaments as tension elements and the microtubules as compression elements, it was a nice, simple, fixed model that really publicized the whole thing and made it much more popular and easy to understand. But of course, as you rightly say, both the microfilaments and microtubules are not static. They're changing all the time. They're treadmilling. So I say this for the benefit of anyone who's not familiar with the cytoskeleton so much. They polymerize at one end, add bits at one end, and they dissociate at the opposite end. And those parts move from one end to the other. So the whole thing is changing all the time. And although it won't fit into whatever the classic Tensegrity model is, because there isn't a classic, apart from what Steve described in the first place with the Tensegrity icosahedron, all these other things are working to understand the details. And now we're getting into this new stuff, which with electron microscopes and the work you do, help us to understand a bit better. But I think it's perfectly working with Tensegrity. I really do.
I don't work with electron microscopy, and I also really admire the people who can do that, especially with two stains, because they separated the tubulin from the actin, and that kind of work is like, that takes a lot of patience.
Yes, yes. And there's a lot of work gone into, probably months, perhaps years of work gone into this sort of our presentation, and there's so much more detail to explore. But I think Tensegrity is
and a big team, a big network of people who also heavily contributed to that work.
Yeah, so many people, lots and lots of time. Yeah. And then on this end, right, a lot of different people studying the body and biology and medicine in the context of the Biotensegrity model. So I think it's fabulous how you can help bring us together, EChO. I want to turn it over to Neil for a second. If I just have a sense, Neil, that you've got a few things to say here.
Well, yes, thank you very much. I was interested, as we think about the metastatic,
Neil, hang on. Can you adjust your mic for us, please?
I'm so sorry.
Mariana will help you, I think.
Or move further away from your mic. I think that's
Okay, is that better? Is that better? 1, 2, 3.
a little further away. Or
Is that
turn your mic down. That's what worked last time.
Okay, move on with someone else while I play with this and we'll come back to me.
All right.
Do you want me to put you in a room with Mariana?
No, I think I'll manage.
Okay.
Okay.
So Susan, do you want me to ask the next question then?
Yeah, but I want to just hear from some of our regulars who I know have been working in this area. And I don't know, Carol, how much of it you heard or what you wanted, if you had any thoughts. I just want to make sure we get to certain people who really have been deep into this field so that we get that angle. Because part of the reason is, this is selfish on my part, this is so over my head and I love it. So Carol, where are you?
I'm underwater with you.
Okay.
But I do want to say, I do want to remind you that well, maybe to restate that, as Steve says, all of you say time and time again, these models are fields. They're not structures inside. They're fields. They're forces. We're looking at forces and the description of how forces are working together within the cell. And so I think Graham was absolutely right when he discussed Ingeber's introducing it to us and bringing it to the public eye, but then the blessing and the curse, people get stuck on that. Is it that it's either tension or compression? And as Steve says, in the body, it's always both, and it's always a product of both. It's never either or. But what what is brought to mind is Mina Bissell's work, and I don't know whether EChO knows about this, but the communication between the cancer cell and the fascia around it, and the recognition that that when a breast tumor is placed in cells of a rat breast tumor are placed in a petri dish with regular growth medium, it grows bigger. But when those same breast cells are placed in a petri dish with normal fascia, the breast cells revert to normal. And when they looked at what might be the mechanism working here, they took out the communication system of the cells with one another and found out that when the cells when cancer cells don't know where they are, and so the hypothesis was they're growing bigger to get a better sense of where they are in relationship to one another and in relationship to the extracellular matrix.
And Carol, will you clarify what you mean by normal fascia, please?
On rat tissue, rat extracellular matrix that was not involved in any kind of cancer process, any disease process.
Yeah, healthy extracellular
matrix from a rat, right?
Yeah.
In a rat breast, normal breast. And the idea was, then I read in this same article, I'd have to dig it up for you, that the scientists from MIT and I think UC Davis were looking at what chemistry could be involved, and I thought about you, EChO, what chemistry could be involved in softening the extracellular matrix so that there wouldn't be a contamination, that is, get rid of the fascial restriction that caused the cells not to be able to talk to one another. And of course, what we teach in the manual therapy that I practice, Barnes myofascial, is that the infrared energy from our hands and the elongation from the hands opens up the tissue and gets rid of the restrictions manually without the side effects of chemistry. and then that really is the more natural way to go about it. And I imagine Susan Findlay might have something to say about this too, because that's her life's work. But that's all I wanted to say. It was a marvelous presentation, and like Susan, I'm just absolutely amazed at what you're doing, having had family members who died from metastatic disease. So this is really very, very meaningful work.
Yeah, and I know, I know we want to hear from Diane and Susan Findlay and Dr. George Roth. Chris, where are we? I know you've been kind of tracking.
Yeah, I have. So we have Neil who wants to speak. George also wants to speak. Diane Woodruff asked a question before George, so that'll leave that up to you to navigate.
And then we want to hear from
Neil,
Susan
perhaps.
Findlay.
Sorry, Susan.
And then we want to hear from Susan Findlay as well, because we know she works in oncology. So
Susan Findlay and Eleanor also has Eleanor Silverstein, is that the correct pronunciation, Eleanor? Also has a question. So that's the summary of the questions. Would you like me to put them in the chat for you?
no, why don't we just, because Carol just mentioned Susan, why don't we go to Susan and then we'll go to Neil and then George and Eleanor. How does that sound?
Don't forget Diane.
Oh, I'm sorry.
She's one of the first people. Actually.
Yeah, let's go to Diane and then Susan Findlay. Let's do that. Okay. Diane, you'll have to unmute.
My question really came from a much earlier tea party in which Neil was speaking, and he spoke about oncologists who did not work with the basement membrane because there was no basement membrane to work with. And my question was, if this cancer goes into remission or is cured, does the basement membrane restore itself? Because there's always a basement membrane, except apparently in this situation.
Echo, do you have a thought on that?
I know that people who focus more on the migration aspect of cancer metastasis think about the basement membrane a lot and that disruption, but they're looking at it in petri dishes a lot of the time. So I don't know the answer to whether. I mean, I guess you would think. Yeah, I don't really have the answer.
But Echo, I really appreciated your slide that showed most of the work is being done here. Most of the mortality is happening here. Here is this huge gap between. So maybe we should go to Neil before we go to Susan Findlay. Do you mind, Susan?
Not at all.
Okay.
Thank you. Is the sound a little better?
Much.
Yes, I was also interested in the basement membrane aspect of this, because of course normal cells hang in the tissues within a structure like washing hanging on a line. And as cells become free to move away and travel within the body, they do so by letting go or by the washing line disintegrating or breaking. Under typical biological circumstances, the metastatic cascade doesn't involve a single cell, but more often clusters of cells. And although I totally understand why EChO is reducing this to the single cell, I think as you become more reductive and look at one cell out of the context of the group of cells, we may be introducing artifacts. And of course, we're very sensitive to this idea because what we learned originally about muscle had to do with muscle strips in an electrolyte bath. And most of what we learned about muscle shortening, for example, was largely artifact of the way that we reduced our view of muscle. So I'm very grateful to EChO for her remarks today, but I wonder whether she would agree with me about some of these limitations when it comes to interpretation.
So what I will say, because I have worked with patient blood and like basically isolating CTCs a bit, there are more single cells, once they get into circulation, there are more single cells than cluster cells. Now, when it comes to which ones reattach and survive, yes, the cluster ones have a very big advantage over the single cells. And and that's kind of what I was showing you in the final slide, is we are starting to look at assays that also consider the clustering potential of these cells. However, in that, the other thing about clusters is non-homogeneous cell types also have an advantage. So when I'm looking at one cell type, when you have it break off, you might not have the sort of, homogeneous population of cells break off is another thing. And there are people who look at that also. There are some new tools that make looking at CTCs and suspended cells a lot easier. We didn't always have the technology to ask those questions, even if we had wanted to. I tried to answer them earlier on, which was kind of nice. But now that we have ways to like separate, because you might, in a really aggressive cancer patient, you might have like just a handful of CTCs out of billions of blood cells. So trying to get that is probably harder than a needle in a haystack kind of problem. Although the one cell or cluster that makes it obviously does a lot of damage.
Can I follow that up with a very quick question about how these outgrowths fit in with what we know about the dendritic cells that are so important universally within biology, but particularly within embryology and growth and development?
So the dendritic cells, and this is not as much my area, those are immunocells, right? Are you talking about like kind of the CAR T cells that are like the latest therapy where we basically train someone's cells to attack the cancer cells and they get a fever for a few days and they like it's worked some miracles in some cases. In fact, I met a scientist who had a terminal cancer. I'm blanking on which one. And he had closed down his lab. He had helped his grad students find other labs. He expected to die from this disease. And he was more on the genetic side and kind of bashed the immunologist a little more. And then he got the CAR T cell treatment and completely recovered. And now he's shifted more to like immunology aspects. I don't focus on immunology. I think they've really done some cutting edge stuff. But yeah, I focus more on the mechanical behavior in the floating environment.
Thank you very much.
So Susan Findlay?
Well, my head's about here with information. I'm trying to tie it into what this would mean as a manual therapist, trying to give it into a language that a touch therapist might be able to find it useful. So I'm not forming a question yet. I'm going to go away with this and try to assimilate it. And then maybe next time I'll have some questions for you, but lovely presentation.
Yeah, it seems like thank you, Susan. It seems like the scale levels are so drastically different. Because at the touch, you're at the kind of macro level of the body, and ECHO's working at this, very, very tiny, teeny, tiny little level. And so that connection, would you agree, Steve, is you don't want to be forcing it. I mean, I think it's great that we can be informed, but there's such radically different scale
I'm just wondering with the touch and compression and the, our energy as well as compression tension. What is that? How is that going to influence what you've done, ECHO, in your tentacles? And is that going to cause damage, or is it actually going to enhance?
Because I know at the last, ECHO, let me just say that the last big that we had in Belgium, there was a bit of a controversy because there were some people saying there's some research indicating that if someone has cancer, you shouldn't be touching them, you shouldn't be massaging them. And there's a controversy in the field. Susan Findlay is a oncology massage therapist. So actually, ECHO, if you don't mind, I'd like to hear first from Steve and then from you. Do you mind?
Sure.
I'm not sure what you want from me.
Oh, I was just commenting on these, how they're drastically different scale levels, or maybe I'm misreading something. The scale level you're at with whole body connection and touch, although of course it reaches into the lower scale levels, versus where where ECHO's doing her research, we may have to be patient about how the connections through are made in terms of her information, or what do you think?
Okay, wait, again, let me go back to 1975 when I first got into this thing. And I started with the spine, but I immediately recognized that I couldn't understand the spine unless I understood from the very first cell and even the formation of the cell. So as I pointed out at that time, there was no electron microscope. This kind of stuff was just not even known. We only had, in fact, we were even short a chromosome in 1957. We were short a chromosome, and in 58, they got another chromosome. Well, I'll tell you that I knew right from the beginning that I had to start back where ECHO is now, in a sense, working on those things. And she shows me these things that differ from a tensegrity, but I see tensegrity right down to the very bottom of the thing she's doing. And so I don't see, so my initial aha is proven to be right, because ECHO's now shown that to me, that I see these things right from start to beginning. Saying that, I pay no attention to that stuff because I'm a physical therapy, manual therapist, and I'm interested in how the musculoskeletal system works. I just have to know this as background information that no matter what level I'm looking at, it's just another level of the tensegrity.
Thanks, Steve. So let's get Eleanor's ECHO's response, and then we'll go to George and then Eleanor Silverstein.
Yeah, I don't know how much you can impact CTCs. I'm not as familiar on the impact of massage on primary tumor, or working with immunocompromised individuals, or the literature in either direction with that. That's kind of more on the clinical side, and I stay a little bit in my lane. But I will say this, the best case scenario is we never get to the point that it metastasizes, right? There was, I probably shouldn't say this on YouTube, but I'm going to. When I was at the NIH, we used to joke that we should be called the National Institute of Diseases instead of the National Institute of Health, because all of the questions were asked like what treatment can we give in the late stage? And there wasn't a lot of support for questions that we could ask in sort of the preventative and earlier side of disease progression. We know that stress increases your likelihood of cancer and aggressiveness. And things like massage therapy help that. So, we're not all always going to be doing all aspects of this problem. And I think, prevention of cancer has a immense value and will always be easier than preventing it from spreading or helping it once it spreads or even as it becomes a primary tumor.
Thanks, EChO. George Roth.
Oh, hi. Good afternoon. It was quite an interesting presentation. Thank you so much. And it's my background is in chiropractic osteopathy, but also naturopathic medicine. So I do see people with more serious illnesses, and over the years, some people with cancer, although I don't specialize in it. I come across this is a question also for you, Steve, because I'm interested in how our microbiome interacts with the extracellular matrix and how that integrates with the communication system that can be disrupted. Okay, so I'd like your take on that. As well, you did talk about the influence of electrical properties, like the electrical influence on some of these microfilaments and so on. And I'm curious about that because that's another area that I'm interested in. One quick thought on the microbiome, and that is, not sure, and I don't know if we just talked about it before since I'm fairly new to these discussions. There's a fellow named Dr. Zach Bush. I don't know if you guys have heard of him, but he's a cancer researcher who looked at the intercommunication between cells and how the microbiome is disrupted by various things like glyphosate, also known as Roundup, and also how glyphosate impacts on the tight junctions between cells in various areas like the gut, blood-brain barrier, and so on, that can lead to increased rates of cancer, loss of communication between cells, as he says, as well, which I think fits in with the Tensegrity model as well. So I'm just curious about the microbiome part, if anyone's looked at that. And secondly, the influence of electrical fields or electrical current on normalization or effects on these structures. So I'd like to hear from Steve and EChO on that.
Great. Which of you wants to go first? You want to jump in, EChO?
Sure. On the electrical, it would be really hard to study that on like floating cells, right?
Yeah.
Like, as well as on like a one cellular level. I mean, they do that with neurons, but doing that with epithelial cells would be really tough. What we do here, the impedance measurements I was talking about, is just you have a plate with an electrical force across it. And the more things that attach to it, the more it impedes that current across. So
and the more it impedes what? Say that again.
The electrical current coming across.
Yeah.
So what we're doing is like we have a substrate down and we're using that as a way to measure how many things are attaching to it.
Oh, I see. Okay. So you're measuring.
So we're not actually measuring the impedance on the cell, which would be really hard to do. I mean, maybe you could do that with some sort of like charged beads. But I actually started my graduate work working like with an optical stretcher. And when you start working with magnetic beads and really focused light, you like cook your cell, you introduce a lot of things that damage the cell and is so far from its original conditions. Those are all just very invasive assays, and sometimes we learn things from it, but we're also not always sure how much that is from, how much that might not be from like the assay that we're using to measure things. So that's something we always, like, all these perturbations, they also change what we're looking at. And so we have to be very careful about what we compare with one group to another group. And that's why we're kind of reductionist, because there is just so much going on at the cellular level. Like, you have to kind of pick one thing to focus on. And then of course you always have to relate it to the clinical level, right?
And I appreciated, EChO, how you said, okay, here it is viewed this way, viewed this way, viewed this way, but then you have to consider this. And you really did try to bridge that kind of gap between the reductionism and the holism, which I think is remarkable at the level you're working on. Steve, what's your response to this?
Well, yeah, my response is that, again, I think a lot of this links into Jerry Pollack's work, where that water element and the electron fields are present at every level, including the water shell around what we're calling water shell around a protein. So it goes down to that level. But when we talked to him, he was also talking about the negative charge across articulate cartilage. So that same kind of thing goes through from the macro to the micro and back and forth. So we have to say that it fits into this thing, saying that I'm a mechanic. I don't deal, when I'm doing this stuff, I don't deal in pathology. I deal in mechanics. I deal in mechanical malfunctions. And that when I'm doing manual therapy, I'm not treating disease, I'm treating a mechanical malfunction. Now, when you get down to cancers and things, then you're treating disease. And I think it's a different story. And I don't like to cross into that field at this point.
Well, yeah, just taking off from that point, I think the challenge with, that was introduced by, glyphosate in the environment and our food supply and the water supply is that basically it's a antibiotic. Glyphosate is an antibiotic. And so we've decimated the microbiome. And if you think of the microbiome as an integral part of the extracellular matrix, which I believe it is, according to some of the recent studies I'm reading, is that it's like taking a part out of this machine. We literally have unplugged an important contributor to the intercellular communication system, which leads to pathology, and I think as mechanical, therapists or whatever we're looking at, it's like there's now a part of this machinery that's been removed.
And
so that's my concern. Pardon me?
And hearing you, George, just flashes on a statement that Susan's made about we are composite beings. But I'm just jumping in here.
Right, sorry.
Because earlier on, Neil commented that he would like to contribute something around easy water. Of course, I think it was just last week we had Gerald Pollack with us. Mm-hmm. So Susan, you didn't know that. My apologies for jumping in.
No, that's fine. Why don't we go to Eleanor, Eleanor Silverstein, who's been waiting so patiently, and then we'll jump back over to Neil for the easy water discussion. And I have to say, this EChO, look at what you've done here. This is so wonderful. You are sparking
things up.
So many fabulous kind of whole entire areas of discussion that you could spend hours on. So here we're doing this kind of flyover. So let's go over to Eleanor Silverstein. Thanks.
Hi, Eleanor. Thank you so much for everything that you said. And can you hear me because I'm on an external mic? Good.
Yeah.
Okay. So I had so many questions. And then, of course, Diane Woodruff brought in the manual part, the touch part, the compression part, the movement, the flow. And Diane said it and Susan said it. And I'm a Feldenkrais practitioner. So, we work through touch and movement and flow. And so I am super thankful George brought this this other part up because I love biochemistry, because that just took care of my second question. Because with cancer, like, I love the idea that we can work within the biotensegral connection and flow within the cells and the matrix between the cells and all of that. But if our biochemistry is constantly being interfered with by external environments, like Mariana said with her beta fish, if we don't clean that up, we're going to be a lot of sick beta fish, no matter how much touch we do. So thank you for that, George. And but here's my question to you then. So that took care of two, unless you want to talk about that. I'm really curious about your thoughts on the spikes that you brought up between the viruses, cancer, and COVID cells. Can you talk a little bit more? Because that's
because we actually, like, there are members of my lab that very early in this disease were looking a little bit at some of that and collaborating with a COVID lab. Ultimately, you know, I think our time was better spent investing in a vaccine. But what's also really interesting, so COVID impacts the lungs, right? And these microtentacles also, like, tends to be the reattachment site that we see, that, like, the assay we do with mice. I haven't done animal work, but there are people who do, and it injects in the tail vein, and they get caught in the lungs, is where the ones with microtentacles tend to concentrate. And you can think of it when you're at this mechanical level, like you're that's where the oxygen exchanges in the alveoli of the lungs. And there's really super fine capillary networks there, right? And so they're maybe more likely to come into contact with the blood vessel walls in those really thin capillaries than other places. But it's also near that site of exchange. And I think that's a really interesting question. From here, I really want to focus a lot more on, like, how having these protrusions help things latch to things. That's a question that hasn't fully been answered. And I think it's, for me, that's like my next that's like my next step, is like growing uniform endothelial cells and saying what makes these things stick or not stick. I do joke that I'm a bit of a caveman scientist, because I like to reduce to simple questions of, like, so do they have more microtentacles or less microtentacles? And the next step is, does it stick? Does it not stick?
Wait, EChO, did you say caveman scientist?
Yes.
Oh, that's great. Thank you.
Well, because I work with people in cell signaling, and I'm always amazed at the work that they do. I mean, they have these, like, cell signaling networks, and they're so complicated. You walk down the halls of NIH, and they'll have posters of it that fill, like, a whole hallway wall of this signal touching that signal. And I don't know how they manage to look at it and simplify it and impact that in a way that just kind of blows my mind. I kind of look at things on the micro level and say, okay, what is this going to do on the macro, on a more, and I say macro scale, and I'm talking about a cell.
Right.
Like for me, I'm looking at things at a macro scale, and for you guys, you're like, this is such a small level to look at things. And it's always a challenge relating it, like making that transition between scales. And it's why it's important that you do work in all of the scales, even if all of us don't do work at every scale.
Thank you. I think we're back over to you, Neil.
I was going to just make the connection back to what we were talking about last week with Gerald Pollack. As EChO described her model, she discussed the vehicle, which was effectively the placebo, the control, and the control fluid is DMSO, dimethyl sulfoxide. And DMSO is one of the most potent disruptors of bound water in that we know. So one of the
when you're doing it at like 1% or 5%, when you do it at 1/10,000th%, you don't see as much of a difference.
Is that correct? Okay, well, I'm very glad that you were able to clarify that.
And that's experiments you have to do, right? So our lab has done tons of controls where we compare normal to DMSO, right? Like treatment. And yeah, like, you know, when I was newer and involved, I like did one at 1%, and my advisor was like, you can't put that high of a concentration for
excellent.
Exactly that reason.
Thank you for clarifying.
But I think, Neil, you're talking about DMSO, am I saying that right, as being a disruptor of the EZ water, that fourth phase of water. So I don't know if you know this work, EChO, the fourth phase of water is a kind of a soft matter phase, an organized, highly organized exclusionary phase of water. This is the work of Dr. Gerald Pollack, and he was with us last week. And so I think what Neil is talking about is the DMSO being a disruptor of that particular structure. Is that right, Neil?
Yes, that was exactly correct.
So I don't know how that work would interplay with what you're doing, EChO, but I think we have found yet another potential 2 hours for discussion.
I mean, certainly if you use like higher concentrations of DMSO, that would be a problem. I think that kind of gets to a point when I was saying, like, you know, studying prevention, right? Like, I think, you know, we know that cigarettes, for example, cause lung cancer, right? We learned in a very sad and tragic example, all the particulate matter from 9/11 and the impact that it had on lung cancer. We know that small particulate things are really, really bad for your lungs. And yet now we have vaping, and it's like we have to reinvent the world. Like, some of us in cancer are looking at that and we're like, do we have to reprove gravity over and over and over again before we just say hitting your lungs with small particulate things is bad, no matter what the nature of those small particulate things are? And so I think, you know, those questions I think are more, those are more for sort of an environmental studies, where you want to see like what are the environmental impacts of the things that you're ingesting, that you're breathing. Those are more for epidemiologists to look at and then make recommendations to the appropriate agencies, and make sure the FDA then does what it's supposed to and takes that guidance and actually make sure that those things are not above levels that are safe.
Yeah.
And that things are labeled correctly. And I think that's the approach to do that. Although we also need to keep in mind kind of the scale and concentrations of stuff. You know, if you have a really tiny amount of something radioactive, that might have a big effect. But if you have like, you know, like a small molecule of like DMSO in like the huge sink of your whole body, that's probably not going to have a huge impact. If you start, you know, guzzling a whole glass of it, I don't know. Like, is the amount of fluids in your body so much more that it's going to not have a significant impact? How are you going to measure what is a significant impact? Like,
Yeah.
I hope you're not going to wait for a 20-year study on cancer, or you're never going to get your answer.
And then that makes me think of systems, right? Where sometimes a teeny tiny little switch in a system can have catastrophic cascading impact across a whole system. So that becomes another question, right? It's not always a linear proposition.
And I would also say that, like, you know, the burden of evidence is very different for medical things than they are for, say, cigarettes. We have to show that it's safe before people can use it. To get restrictions on those other things, people have to show that it's unsafe, right? Which then makes our work a lot harder, because there are all these, you know, they're like cigarettes and certain percentages of chemicals released into the water or whatnot. And those may develop cancer later on. Like when you look up for DC for water, I think there are like 11 carcinogens in the water supply. Most of them you can filter out. One or two of them, not so easily. Maybe we want to have less of that. But I think maybe pushing some of that burden to figuring out what causes cancer, and making sure the companies that are producing those things have some restraints.
Thank you. You know, I know we're way past time, and I just want to make a note before I check in with Chris. I believe this marks a bit of a milestone for us on the Biotensegrity party, because I believe that EChO is the first senatorial candidate that we've had with us. She did run for senator here in DC, and I voted for her. Are we not supposed to say that?
Because I have given a presentation with my with a government logo.
You can't say anything about it.
I can't. Yeah.
Oh, I hope it's not horrible that
So like in another meeting sometime where I have not given a presentation under the logo, but right now I'm doing this as my scientific work, and
absolutely
I'm supposed to not
jump in, EChO. Just say that
you're not bringing it up, EChO. I am, but thank you. Go ahead, Chris.
I was just going to say I'll jump in and say we'll ask Lisa to edit that out if you wish. We'll talk about that later.
Thank you. It's really important that people understand that I do this work for cancer, for treating cancer, and that when I am in the lab, my thought process is more about, does this experiment support that or not? And that is all that happens in the lab.
You don't bring any political agendas with you into the lab.
You leave that at the door. Now, there are political agendas that decide what we fund and don't fund, and what questions that scientists have the privilege of asking in their labs. But when we go in there and we do our best to pull the stories of what's happening out, we leave that at the door.
Right. So it's about the thing you're studying talking to you. What is this material telling me? What am I getting from them? And you're not putting anything on it. You're trying to leave that behind so that you can get the clearer picture.
And to an extent, because I mean, when you do epidemiology studies, of course, there are disparities.
Yep.
There are some populations that have historically been subjected to extreme injustices, who we've not included in this process. And so they continue to experience those disparities. And so it is really important, and that really shouldn't be political. It's gotten really complicated in the last few years because science tries to stay out of politics. But when politics forces itself on you, I would hope we come back to a point where, like, facts are facts, and like if there are social injustices that impact the epidemiology, that's not something that shouldn't be anything political. Like, we have to include everyone in these efforts.
EChO, I want to thank you for your work and for your compassion. Chris, where are we?
Oh, well, I also want to thank you, EChO, and I look forward to continued discussions with you. We're at our sponsors. Graham, are you ready to do the sponsors?
Yes, thanks, Chris. Our Biotensegrity Party is an all-volunteer production of the Stephen M. Levin Biotensegrity Archive, and made possible through the generous support of our sponsors. And for all those of you watching on YouTube, just want to point out that if you go to the Biotensegrity Archive and register for the free newsletter, you'll notice that there's discounts from all our sponsors for what they have to offer. Handspring Publishing, who produce a wide range of authors and books available for movement and manual therapy professionals. Embodied Biotensegrity, who provide an excellent resource for those of you looking to learn more about biotensegrity. Artifact Pro in Madrid, Spain, who produce wonderful tensegrity models. And International Biotensegrity Congress, which I believe is coming from Brazil, heads up for April the 23rd to the 25th. And we'll find out more as we get into the next few weeks. Thank you. Back to you.
Thank you, Graham. And I will just add that next week, same time, same place, we will have Leonid Bloom speaking about the unreasonable effectiveness of light touch. And we're very excited to have him here. Those of you who have scars, adhesions, and the biotensegral body, you can read Leonid's chapter and do some prep work. We're very excited to have him presenting that work here at the Biotensegrity Party. Okay, Susan, I'm going to pass it over to you to invite a toast.
To invite a toast.
Well, I don't know if you've, I'm not sure if you've arranged for EChO to
EChO, do you want to do another toast?
Oh, I can pass this one.
No, that's fine.
By the way, my cat Mina says hi.
All right, to all of our animal friends.
Yes.
You never get enough credit.
Oh, cheers.